Dear all, a very good day. In this class we are going to look at the electrodes that are used in biomedical instrumentation. So electrodes are a device that converts ionic potential into the electronic potential. Okay. We have in the previous class seen that how ionic potential is generated in a cell. Now for getting some meaningful information from those ionic potentials, we need to convert those ionic potentials into electronic potential. That is where the electrodes come into play. So electrode potential or electrode theory. The interface of metallic ions in solution with their associated metal results in electrical potential that is called electrode potential. Okay, so when when the electrodes come in contact with the ionic liquids, they generate a potential which is known as electrode potential. This potential is a result of difference in the diffusion rates of ion in and out of the metal surface. Equilibrium is produced by the formation of layers of charge at the interface. Similar to the process where there was an equilibrium produced in a cell, in the same manner equilibrium can be produced over the electrodes as well, which is clearly shown in the diagram as we can see below. Okay. These charges are double layer, with the layer nearest to the metal being one of the polarity of one polarity and the next layer of the another polarity. So we can see this is an ionic liquid in which we have inserted an electrode. So on the surface one type of potential has been generated, that is on the surface of the electrode a typical type of potential is generated, whereas the other type of potential is generated in the liquid. This results in the flow of charge carriers or current. Okay, so this charge is double layered, with the layer nearest to the metal being of one polarity and the layer next to the solution being of the opposite polarity. Non-metallic materials such as hydrogen can also have electrode potentials. Okay, and they are also used for the similar purpose. Now interface with the associated ions of the solution. Okay, so non-metallic ions also have a interface with the associated ions in the solution, because they can also result in ionic potentials, like hydrogen have an ionic form in H positive. The electrode potential of a wide variety of metals and alloys can be used. Okay, now it is impossible to determine the absolute electrode potential of a single electrode for measurement of the potential across the electrode and its ionic solution would require placing another metallic interface in the solution. Okay, so what does this statement means? This statement means that using a single electrode it is not always possible to measure the potential. Therefore we need two electrodes, where one will serve as a reference electrode and other will give us the relative potential. Okay, so that is what has been described here. Therefore all electrode potentials are given in a relative value and must be stated in terms of some reference value. Okay, so what it means is that all the electrode potentials have a relative value and they need to be measured against some reference value. Okay, now it is an international agreement that a normal hydrogen electrode should be chosen as a standard and having an electrode potential of 0 volt. That is, by international agreement, hydrogen electrode has been chosen as a reference electrode and its potential is considered as a 0 voltage. Another source of electrode potential is unequal change of ions across a membrane, that is semi-permeable membrane, to a given ionic when the membrane separates liquid solution with the different concentration of that ions. Okay, so the source of electrode potential can also be ionic membranes or semi-permeable membranes, as we have seen in the previous case. For the calculation of potential across any membrane, therefore, we require an equation which has been provided by Ernst and is known as Ernst Equation. The equation is given here, which is given as the potential E is equal to minus RT upon NF, natural log of C1 F1 upon C2 F2. The different parameters in the equation are explained as follows, where R is our gas constant and its value is given as 8.315 into 10 to the power 7 ergs per mole per Kelvin. Okay. Now T is the absolute temperature, N is the valency of ions, F is Faraday's constant, C1 and C2 are two concentrations of ion, and F1 and F2 are respective activity coefficient of the ions. Okay, these values vary from ion to ion and therefore should be used very properly. Now, based on these biopotentials, there are three types of electrodes which are generally used. One is the microelectrode, skin surface electrode, and finally the third one is needle electrode. Okay, a brief has been given here and we will be discussing them in the following discussion. Microelectrodes, they are normally used for the measurement of BP near or within a single cell. Thereafter, skin or surface electrodes are used for measurement of normally electrocardiogram, that is ECG, and EEG, that is electroencephalogram signals. Okay. Then needle electrodes are used to penetrate the skin to record EMG potential from a local region of a brain, or EMG, that is electromyogram, normally used for muscle cells. Now in each case an electrode potential is developed across the interface proportional to the exchange of ions between metal and electrolyte of the body. This means whenever an electrode is used, it interacts with the ionic liquids of our body and an exchange of potential takes place between the electrodes and the ionic liquid, which results in the proportional potential being generated. The double layer acts as a capacitor, thus the equation circuit of a volt consists of a series in resistance and series network of resistance and capacitor. This series network is shown below. Okay, so again in each case an electrode potential developed across the interface proportional to the exchange of ions between the metals and electrode body. The double layer acts as a capacitor. Okay, since there is a double layer of electrode, so it acts as a capacitor. Therefore the equation circuit consists of series with a resistance capacitance network as shown here. Okay, there is a resistance network here and there is an RC network here which is to be considered for the body electrodes. Okay, now since the measurement of blood pressure requires two electrodes, the voltage measured is really the difference between the instantaneous potential of the two electrodes of the same type. Okay, so if we are using two electrodes of a same type for measurement of blood pressure, so difference between them will be the instantaneous. So blood pressure which is being measured is actually the instantaneous difference between the potentials which is observed at the electrodes. Okay, now the DC voltage due to the difference in the electrode potential is called electrode offset voltage. Okay, so the voltage which is generated on the potential is called electrode offset voltage. Okay, now here is a measurement with the help of two electrode system. We can see where each of the electrode has its individual setup of RC network and resistance network, and potentials are applied through the body fluid, and difference in the potential of these two liquids is used or it is required for the measurement of two electrodes. Now coming on to microelectrodes. Microelectrodes are electrodes with the tip sufficiently small to penetrate a single cell, okay, in order to obtain reading from within the cell. This means the dimensions of the needle head of the microelectrode is very small. It is so small that microelectrode can penetrate a single cell in a human body. Okay. Now the tip must be small enough to permit penetration without damaging the cell. It is very important that the cell should not get damaged during the process. Okay, so microelectrodes can be basically of two types: metal, that is electrolytically etching the tip of a fine tungsten or steel wire. Normally either tungsten or steel wire is used. Now it can be also made up of glass in a micropipette form, okay, where glass is drawn into a suitable size. Okay, thereafter this is a figure which shows the commercial microelectrode needle which is available. It has a gold plated pin conductor and a resin insulator. It is basically showing a glass one in which there is a glass drawn mode and a metallic thin film. Okay, then coming on to body surface electrodes. Now body surface electrodes are used to obtain bioelectronic potential from the surface of the body. So these electrodes don't travel too deep within the human body. Okay, these are larger for ECG, that is electrocardiogram, for the measurement of Hertz waves or Hertz signals, and these are very small for EEG or EMG, okay, which are used respectively for the measurement of brain waves or muscle activity. Now there are different types of these electrodes like immersion electrode, plate electrode, floating electrode, suction implant electrodes, or spray on electrode, disposable electrodes, scalp or surface electrodes. Then comes needle electrode. To reduce the interface impedance and consequently movement movement artifacts, some electrodes encephalographs use smaller subadrenal needle to penetrate the scalp for EEG measurement. So these are basically used for measurement of electroencephalogram or brainwave signals. Okay, these don't penetrate much deep, and this is what it looks like, graphical representation. Needle electrode for EMG consists of fine insulated wires placed so that their tips, which are bare, are in contact with nerve muscles and other tissues from which the measurement is made. So these type of needle cells are directly in contact with nerve cells or nerve muscles which we can take signals from. Okay, the remainder of the wire is covered with some other insulating to prevent shorting of the shorting of the needle electrode. It is very important, otherwise they may hamper with the brain signals. Okay, this can be of unipolar with single wire inside the needle, or bipolar which consists of two wires inside the needle. Okay, in the next class we will be discussing about biochemical transducers and its few types. Okay, please remember to revise this part. Okay, thank you.